Hello, I'm Les Bennett, representing the American Association of Pharmaceutical Scientists, and we have the honor, or I have the honor, of carrying interviews of distinguished pharmaceutical scientists. The association wants to put together a video library of individuals who had made significant contributions in the pharmaceutical sciences over the years, and I have the opportunity of serving as that interviewee. So today we are very pleased to have with us Dr. Vinod Shah, who has spent a number of years making a number of outstanding contributions. He is a former president of APS. He was a former individual at FDA who has made a number of significant contributions leading to guidances, which we'll get into. And I always like to point out that Vinod is the highest cited is the highest cited FDA scientist over the entire FDA, and I think that's a great honor. So Vinod, we're very happy to have you here today and to share our platform here for our young scientists and others who were interested in your accomplishments.
Well, I'm also happy to be here and to be a part of this video library, and hopefully the younger scientists, newer scientists will benefit from this interview that Professor Bennett is conducting today. I'm happy to be here and honored to be here, Les.
Great, Vinod. So why don't we start off with a little of your history, you know, what your education was in India and then the U.S. and, you know, let's talk about the reason that you wanted to become a pharmaceutical scientist that led to this history.
Okay. I'm a pharmacy graduate from Madras University. I graduated with a gold medal. I was #1 in the whole university when I graduated. And after that, I wanted to come abroad for higher studies. So I applied at few universities, including San Francisco. I got admission everywhere, but I selected to go to San Francisco primarily because the people told me the climate is much better in San Francisco rather than going at different places. And I came to San Francisco. I did my graduate work with Professor Roger Ketchum. And after finishing my graduation in pharmaceutical chemistry PhD, I decided to go back, primarily because I wanted to go back to my country and serve there. I went back. I was there for about 5 years, but I was not very happy for one reason or the other. And then I decided to come back to San Francisco to do the postdoc. I did my first year of postdoc with Roger Ketchum because that was easy for me to do. And after that, I did about 5.5 to 6 years of postgraduate work with Professor Sidney Riegelman. And that's where I got all my knowledge of pharmacokinetics, biopharmaceutics, and all those areas, which I enjoyed it very much. That's where I had my first interaction with Professor Bennett around that time. And after that, though I had an assurance from Dr. Riegelman that he will have a job for me as long as I want to stay there, but I decided to look for a permanent job opportunities, and I ended up with Food and Drug Administration. I worked that for 30 years as a pharmaceutical scientist, and then I retired from there. That's my history, how I came to San Francisco and did my work.
Okay, Vinod, so that's really nice. So give us a perspective of what divisions and, you know, how you progressed at FDA in terms of getting into the influential positions that you really came about.
The moment I entered the FDA, even when I went there for the first interview, I got the impression that I'm already hired at the FDA. That's how Bernie Cabana, or I should say Dr. Bernie Cabana and Dr. Skelly and Mr. Jin Knapp, they treated me. Like I'm already there and what are my assignments. I started working in the lab because I was working with Dr. Riegelman in the laboratory, and I had indicated I would like to stick with the lab and do the experiments myself. So that's how it started, but then they needed somebody who is a lab scientist, but needed some more interactions with the people in the reviewing division, because they wanted the interactions and face and address the issues and the challenges and all. So I started doing that, and since I was a lab person, they assigned me the dissolution work across all the laboratories in FDA laboratories in United States. To start with, I had about 13 labs. Also, when I was there, they assigned me as a project officer to do the contract research work. So I started doing the research work, had the several different laboratories, and then I gradually grew up into doing the review work, helping the reviewers, and also doing the lab work and doing the contract research work. With all these activities from the biopharmaceutics laboratory, I came down to the headquarters and started working as a branch chief of a science management policy branch, and that's where we started developing all the policies. This is in early 80s with respect to the biopharmaceutics area. Before that, when I was still in the laboratory, we started writing the FDA regulations, the 1977 FDA regulations. Quite a bit of that were written by me and the other groups of the people working over the weekends and days and nights and trying to develop the FDA regulations. So those things really helped me to gradually grow up into more, I would say, type of a position which helped me to really work towards the policies, make the decisions. And when I was the branch chief of a science management policy branch, that again further helped me. And then after that, I had several research contracts with me, which again helped me to continue in my research efforts, working with the different universities and then trying to move forward with that. And then gradually I was the acting associate director, acting director for the different groups and all, and finally moved into the Office of Pharmaceutical Sciences, which was headed by Dr. Roger Williams when he came there. And then we started writing all the guidelines, because those things needed to be written down. And those are the steps, which brought me to the level where I am. And then after several years, then I retired from the FDA.
So one of the things that I think AAPS is most proud of, and I think you are really one of the people that made it happen, is the joint workshops that were held between AAPS and FDA that led to these consensus ideas that then I think turned into the guidances.
Right. Well,
want to talk about that a little bit?
Sure, I can talk about it. That happened in about early 80s. We started saying that industry is on one side, regulators are on the other side, and the academicians are the third part of the issues. And we need to bring all these people together to advance the science. We decided that FDA should make the rules and regulations based on science. And with that intention, we had the workshops starting for when AAPS was not there, we had carried out 2 workshops along with APHA and the other organizations. But once the AAPS was formed, we interacted with the people, management of AAPS, and said that we would like to use AAPS as a neutral platform where the scientists from the different regions can come together, work together, and start developing the policies. So the major first workshop that we did with AAPS and industry together was the workshop on the bioanalytical methods validation, which finally became known as the Crystal City 1 workshop. It was that workshop which resulted in the first development of the bioanalytical methods guidelines. That is only one example. We had similar workshops on other areas, pharmacokinetics, pharmacodynamics, and toxicokinetic measurements, which also resulted in similar papers working with the center director at that time, Dr. Carl Peck. That's where we developed this. And if I can say, I am fortunate to say that in several of these key workshops, Dr. Bennett, you have been part of that. Actually, we started even the very first workshop, I think, where you were involved in the controlled release preparations, and which ended up in the guidelines of the controlled release preparations, how those preparations has to be done. And we were fortunate enough, I would say, that for all this workshop, whenever we had approached any individual, including Professor Gerhard Levy, he was always willing to come and participate whenever there was a need for his expertise to be involved into the workshops. And that was part of the reasons why we were successful enough to achieve what we have been achieving it. And I would say it's partly because maybe the way we were approaching the faculty, the way we had said that we are going to be establishing a neutral platform, we will write the report which will be completely agreed upon by all the speakers and participants and all, and that was the final result, I would say, why we have been successful enough in establishing this. With except the first 1 or 2 workshops when AAPS was not there, we had carried out with the other organizations. But once the AAPS was established, again under your leadership, we continued flourishing and doing the workshops very nicely, and that have resulted in ultimately the guidelines. I mean, the bioanalytical methods, the topical workshops, the SUPAC workshops, which all resulted in the SUPAC guideline, scale up and post approval changes, immediate release, modified release, semi-solids, and all that, and it has been very successful to achieve those levels because of the neutral platform that AAPS had established.
Yeah, thank you. That's good history. So let's talk about certainly your most cited paper, because you are a co-author of the BCS 1995 paper with Dr. Amidon. And give us a little history of, not the BCS paper, but the science behind BCS. Give us a little history of that.
Well, I would say that around in 1990s, Dr. Gordon Amidon came to FDA on his sabbatical. Well, when he came on sabbatical, I don't want to say he was assigned to work with me, but it was in principle that was the case, because each scientist, when they come, they need to be assigned to somebody else to work and interact with each other. And when Gordon came, they said, you work with Vinod. So we both were working together, let me put it this way. We had a research contract with him, and the whole idea was, how can we simplify the bioequivalence requirements? Right now we are requesting the bio for practically every dosage form. That's what came up after the 1984 Waxman-Hatch bill, that any product that wants to come as a generic product after the patent expiration has to do the bioequivalence studies. So at that time our center director was Carl Peck, and he said that we need to change this. There are a lot of drugs where we don't need to carry out this extensive bioequivalence studies. So think about doing something. And at that time Gordon was also working with us to tackle some of the major major dissolution problems, like carbamazepine was one of the drugs which had a major dissolution problem, and how to do that, how to solve that. So, when Gordon and I were working together, we came across so many different issues, how we can put the things together. The BCS actually, as you know, is a combination of the solubility and the permeability. These two factors at least are the old information that we have. It's not something new, but the idea was how can we put these factors together so that it could be more applicable and people can easily understand what's happening into the body. At the same time, we had a research contract with Gordon Amidon, which was again further given it also to Hans Lennenas, who was doing the permeability study, in situ studies on in the human subjects. So we tried to put all these factors together and we came up with the so-called the biopharmaceutics classification system, and that was the basis how of all our interactions working together, hours and hours of discussion that resulted in the formation of the BCS. So BCS then it says, okay, we can take care of the solubility, we can take care of the permeability, but normally we don't take the dose the API, the active principle itself. We have to modify that, we have to formulate that and make it into a tablet or a capsule or a dosage form. That's where then we introduce the dissolution again, and we had the different characteristics of the dissolutions, and that's how the BCS came into the picture of in this world, and it has become a very common name now. It's very common, but I would say that after couple of years of the BCS, we are also working from the FIP side with the WHO. So the feeling was the criteria for the bio waiver using the BCS by the FDA is very stringent, very hard, and it needs to be slightly loosened up. And we did that using the scientific principles that was well established with the BCS and the dissolution knowledge that we have gained to slightly make it easier for the class 2 and class 3 drugs, and that's how the BCS has been quite successful in moving forward.
So your impact has been, you know, we talked about the bioanalytical. That was an unbelievable impact because it provided the guidance for everyone of what was the rules, and everybody agreed. I mean, we had this consensus. And then I think people view you probably more from the dissolution and the oral bioavailability because of BCS, but your impact in terms of alternate dosage forms has also been extremely significant and important. And give us a little perspective on that.
Okay. Maybe I can go back a little bit. When I was doing postdoctoral research work with Sid Riegelman, we were looking at the skin as one of the organs and how the drug is being transported inside the skin. Actually, with Sid, we were the first one really to demonstrate that you take the drug orally, grisofulvin, it comes through the sweat inside the front of the body within 8 hours, and it gets deposited into the skin. That's how we were able to identify and measure the quantitative determination of the drug in the skin. But then when I came to the FDA, the first task assigned to me was to determine the bioequivalency for the topical dosage forms. So then we started doing the research work in terms of the bioequivalency. How do we do for the topical dosage forms, and that's what we have been working on. At the same time, we had lots of challenges for the oral drugs which were given, because some people were claiming the product is not studied in the patients, and therefore even though FDA approves it as a bioequivalent, maybe it's not equivalent, it has this problem, that problem. So to solve some of those things, we had also had a research contract with Dr. Marvin Meyer at University of Tennessee, and before that with University of Georgia, to study the bioequivalency of the marketed dosage forms and see whether there are any differences or not, and that's how I got involved also into the oral dosage forms. And then with respect to the more impact on these things, we needed to develop the guidelines for the oral dosage forms. So we developed the first guidelines for the oral dosage forms, how to do the bioequivalence studies, what are the requirements, what needs to be done. Of course, to conclude that we have the bioanalytical methods. But in order to do that, we had to conduct the studies, how to conduct, and also all that information was gathered, captured, put together from the research studies that we were having it at different universities, combine it together to come up with this. And that's how I ended up in now, as you know, doing significant presentations in terms of the oral dosage forms, and also still involved in doing the research at different universities, even though I'm not at the agency, but still working to develop the good methods for the topical dosage forms.
So let me switch gears. I'm going to come back to, why you retired, but I want to switch gears and talk about, as an Indian pharmaceutical scientist, you're wearing your AAPS fellow pin, and I know you were not the first Indian pharmaceutical scientist to become a fellow, but you certainly were the first Indian pharmaceutical scientist to be president of AAPS.
That's true.
And I think it'd be worthwhile for your colleagues, both from India and from not, because there certainly was some concern at an earlier time, that the Indian scientists were not being adequately represented and recognized within the association, and yet I think you're certainly the leader and the person that's really been the groundbreaker. And give us a little, can you give us some perspective on that, of your view of that over the years?
Well, that's true. Like, I had some difficulties also when I was approached by quite a few friends that maybe, since I have done quite a few of the workshops, I had been also in the AAPS executive councils, had started one of the pioneers in developing the RS section when AAPS got rearranged and all that. They said, it's time for me to run for AAPS president. I said, no, I don't think I will win that, because if you take a look into the history, there is nobody from the FDA who has become the president of AAPS. Most of the presidents are from the academic institutions. There are some from the industry who are at very high positions, and I don't have any of those positions or things that I can say which will back up into me. But the friends said, no, you still have enough background and enough recognition in the pharmaceutical field among the pharmaceutical scientists that you should try to run for it, and let's see what happens. And I did run for the AAPS, and as you know, I did not win for the first attempt. I did not feel bad at all, because I know that I lost it against one of the well-known professor. So I did not feel bad, but later on again they asked me to run it, and I did win. And outside in US, at times whenever I have been to India and all, and I have been recognized as the first Indian to achieve this status of being the president of AAPS, the most premier pharmaceutical scientists association in the world. I mean, it is still, and I'm still being recognized that way, and people here at the AAPS, whenever they meet, they do feel that yes, there is a possibility that you can, I mean the other Indians can also gradually go up to this particular level. And so I feel proud about that being the first Indian to become the AAPS president, first FDA to become the AAPS president. So I feel proud about that. So,
do you think? I was trying to get a little touchy there. You know, do you think in those early days that there was sort of a discriminatory position or something, and that's abated over the years, or do you think
I
that's
would
just
not think so. I don't think so that because I never had any feelings at any time at any of the stages of AAPS, like I was there, I became when the AAPS was formed, I was, as you know, sustaining member of the AAPS, and I still have that certificate signed by the first president of AAPS, Les Bennett. I still have that. Actually, even the AAPS members staff were not aware of that. There is something like a sustaining membership. And I made a copy of that and showed it to them, and then they realized it. So starting even from that, any time I had to do any activities, either programmings, workshops, again, like you can say that I was an Indian trying to be in all these different areas, but I never had a feeling that the Indians are put down. If you have the credibility to do the activity, you are always supported by that. I had been always supported by almost all the staff of AAPS and everywhere else. So I would not say that there was that, but some people may have that feeling, but I never had that feeling that Indians were not well recognized if they have the credits.
Good. Okay, that's good. I thought it'd be worth at least
No,
talking about.
that's perfectly all right.
Yeah.
And actually, if you see it, more than 30% of the people attending the meetings or trying to make the posters, presentation speakers, they are all Indians. You see that from Indian origin. So if they have such feeling, I think they should go back and say why they are having that. If they are worthy of doing better, then they should be able to come up and move forward with
Well,
that.
you're certainly an example of how successful you can become, and I know everyone views you with great respect for that. So let's say, so you spent, and you've mentioned it twice, you spent your 30 years at the FDA, and then you decided to retire, and give us a perspective there, because you were well recognized at the FDA and certainly a leader in the field. So
yes, when I was at the FDA, yes, that's true, I was well recognized. I was doing a lot of travel again at that time. After the agreement with my management, I was doing the travel for AAPS, for FIP, and the different organizations, taking my own time. Even when I was the president of AAPS, we had a clear understanding that any activities I do for the AAPS has to be done on my own time and not on the time of the FDA. And at that time, when we had to do some workshops and some meetings, at least again it was all done on my own time. I was with the FIP. I was elected as a scientific secretary for the FIP. Then my travel and everything started increasing, and then more people started recognizing and requesting me to participate and develop more workshops. Well, all those things always has to be done on my own time. I had not enough time to do the work of the FDA and also to do this work together combined. And I felt that having at the FDA for 30 years, I have learned enough and I have given enough of my knowledge to the agency. Now people outside are still in need of some of the knowledge and the things I have. Maybe I can just go out and try to do that. So after 30 years, I decided that okay, it's time for me to leave the agency and still continue doing this outside activities, meetings and all as possible. Actually, when I left the agency, I was being identified as the ambassador of the FDA. Even though I was not with I was working outside. Even afterwards, people still recognize and see me as an FDA individual, even though I tell them I am no longer with the FDA. But still they say that they are able to get more information and more interactions with me about the requirements, rules, regulations, what can be done, what cannot be done, talking to me rather than talking to anybody else over there at the agency. So I tell them, if you are happy with it, I am happy too, but please remember, I am no longer with the agency.
So, Vinod, so
sorry,
as the
I moved away slightly from
that's
what you were looking.
You were the scientific secretary of FIP for 6 years, but
8 years.
Or 8 years, sorry. But I think you are recognized as the individual in the world who has been the most influential in providing these workshops and guidance to third world countries and second and first world countries throughout the world. I think you are the person that has done that, and every one of us who have participated and benefited from it really are appreciative of you, and it's been a driving force that really has been so important for pharmaceutical sciences throughout the world, but also I think important for you. So I'd like to hear why you felt this is important. I know you still feel it is. I just want to give you the opportunity to mention it.
Well, as I indicated, I had picked up enough knowledge after working for 30 years at the agency, and then I was with the FIP, right? So I felt that I'm not able to gain anything more by still working at the agency. And I really enjoy trying to talk with the people, trying to provide the knowledge, whatever I have, to the other people who needs it. Actually, you can say that they're all hungry to talk to us, to get the information from us, like all the workshops we do. They are so eager. You have seen that. And even in India and the other countries where some of the times you have not been there, but I have given so many presentations, people just want to know more and more, and I felt that they still would like us to spend more time with them, gain more knowledge, at least it is helping them. And those are the things I feel it's good for me. We have learned enough, now it is time for us to give it to the others who need it, and that's the way I feel about it. And in order to participate more into these kinds of outside activities, I had decided to leave the agency and do that.
So it's also been, your ambassadorship, as you said, has really been important for the AAPS, because AAPS' goal to assist pharmaceutical scientists throughout the world, and I don't think there's anybody in the world that's done more than you, and congratulations.
Well, thank you.
And I think it's been really important, and we've all benefited.
I would say that it is possible because of the support from AAPS to start with, support from FIP, and also support from the people who are able to go along with us, together for these types of activities and the things. One person alone cannot do that. I can just do only 1, 2, or 3 meetings, and sometimes I have given up to 4 or 5 lectures. Why? Because the other people promise me and then they don't show up, and then I end up in doing it. But now I have few people who, once they say yes, they are always there supporting me, and I think that's what helps really to move forward with that list. Without that, it would not be possible. Yes, I've been able to do this, and I think I have already indicated before, it's because of your strong support to me, Les, that has helped me really to move more forward, more and more in these areas. So I really want to thank you too, Les, for being out front and telling me, yes, let's do it, we can do it, sorry you cannot come, but you will provide a video. So all these things helped me to move forward with the goal that we have is, at least I have, is to improve the drug product quality in the developing countries. How can we do that?
Okay, so I'm going to go back to the FDA in terms of not so much you're working at the FDA, but your view today of what's going on in terms of the FDA and the science and stuff, I have a positive view. I think pharmaceutical, I'm thinking more of pharmaceutical scientists within the FDA, having influence and bringing about good change.
Well, when I was there, we started saying that we need to set up our regulations and policies all based on science, our requirements based on science. With that view in mind, I think we have progressed quite significantly on that. Office of Pharmaceutical Sciences was created with that, and the science was the key focus on that. The same thing had happened to the Office of Generic Drugs. Science was the focus of that we need to establish the requirements, the guidelines and everything based on science. But even when we do that science, we needed to make sure that the medical group of people, the doctors, the MDs and all who are in the different groups, they are also in line with us. It cannot be that they are completely separate. So we had done that. We had talked several times in several our meetings also, like Dr. Bob Temple has been participating in our meetings and really providing us the medical support for our policies wherever we are moving forward. And then in a similar situation at the OGD, Office of Generic Drugs, a new science division was established which was headed by Lawrence, and he started also to have lot of science input to build up the science before any guidelines or the policies are developed. So again, that was based on the science he was doing it. So I would say that based on that, the things are moving in right direction. They are moving forward. They have formed several different committees and the groups to discuss the latest issues, like the BCS, going back to that, can we give a bio waiver to class 2, class 3 drugs, what not, and all those things have been discussed with the scientists from the new drugs as well as the generic drugs together in order to move forward in the right direction. So I would say that overall, the picture wise, scientifically, pharmaceutical science wise, they are moving forward. At least that's the view that they have. The same thing with like Dr. Woodcock has now is going to be establishing the Office of Pharmaceutical Product Quality, OPQ. Well, again, the whole goal is we need to have the product of better quality. How we do it, what not, that's a different story, small nitty-gritty things and all, but overall the goal is to have the product of a better quality into the market. And that's the same goal like we have. How can we improve the product quality in the developing countries, the third world countries, you know what we call it. And that's why we do the workshops. We try to bring them up, provide the knowledge. We also do like the dissolution is the most important one. We have done lots of workshops where we do show hands-on dissolution workshops under the FIP leadership, and we have done that too. So the whole thing is improving a better quality for the patients.
So as this, you won a special presidential citation at this meeting of AAPS, and I think it really reflects the activities we've just been talking about at the international level and representing AAPS at the international level. Do you have goals yourself in terms of what you want to accomplish in the next few years? In terms of things that beyond what we've been doing?
Yes, I would say that I still have some goals to accomplish that. This interview is for a distinguished pharmaceutical scientist.
Good, all right.
Right?
Yeah, that's
so
right.
that's the goal I would like to
be distinguished. Okay. So Vinod, thank you so much for sharing your time today, and we, no, don't stand.
Okay.
And we greatly appreciate.
Thank you.
We love to share and to hear your view, and it's been our pleasure to have one of the distinguished pharmaceutical scientists share our platform with us today. Thank you very much.
Thank you very much. I appreciate the opportunity I had to share these views with you, Les. Thank you very much.
Stay seated.
Okay.